Hey everyone, and welcome to another episode of AMiNDR. My name is Heather, and I'm your host for today's episode. I'll be summarizing the papers published in August 2020 that examine the role of glia, neuroinflammation, or just generally the immune system in Alzheimer's disease.
Welcome to AMiNDR, a podcast where we summarize the latest publications on Alzheimer's disease for you, so you can spend more time doing awesome research. For every month, you'll find a series of episodes by theme, and each comes with a bibliography. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast useful and accessible.
This is a topic that I'm particularly excited to talk about as I'm in the final leg of my master's program in neuroscience, where I'm looking at the role of neuroinflammation and microglia. So super relevant to today's episode. I think just generally, glia are starting to get a lot of attention in the neuroscience community. And as labs start to investigate glia in the context of Alzheimer's, there's been a lot of really cool advancements in our understanding of the disease. So I think today's episode is super topical, super relevant. There's going to be 15 papers that I'm summarizing today, which I've divided kind of broadly into 3 different categories. So papers about astrocytes, papers about microglia, and papers that are more broad to just neuroinflammation and the immune system. Of course, there's always some overlap between all of these things, but we've just kind of parsed it out for your convenience. I'm going to start with the first 3 episodes, which are more specific to astrocytes, and then move into the bulk of today's episode, which will be the microglia research. Then we'll finish off with the papers about neuroinflammation and the immune system. If one of these categories is something that's more relevant to your own work or you're just especially interested in, totally feel free to skip ahead to the topic of your choice. And then, of course, for any of these abstracts that I'm summarizing today, if you're more interested in reading a bit more in depth about it, be sure to check out our bibliography, which you can access through the episode notes, and we'll have each paper timestamped for your convenience so you can go back and listen to it again. Okay, let's talk about astrocytes. Okay, our first paper is titled Proteomic Analysis Links Alterations of Bioenergetics, Mitochondria-ER Interactions, and Proteostasis in Hippocampal Astrocytes from 3x TG80 mice. First author is diminishes, last author Pavel, and this study was published in the Journal of Cell Death and Disease. So this study used immortalized hippocampal astrocytes from the mutant 3x TG80 mice to look at the interaction between astrocytes and mitochondrial dysfunction in a model of Alzheimer's. The authors found that there was an impairment in bioenergetics, so meaning that glycolysis and mitochondrial oxygen consumption were reduced. At the same time, they also found that these astrocytes showed an increase in the production of ROS, or reactive oxygen species. The authors then performed proteomic analysis on a mitochondria ER-enriched sample from the astrocytes, and they found that there was no alteration in the expression of mitochondrial and OXPHOS proteins. However, proteins related to both general ER functioning and protein synthesis had a decreased expression. Since astrocytes are also critical for calcium homeostasis, these authors also used an ER and mitochondria-targeted calcium probe. They demonstrated that the ER in this line of astrocytes is overloaded with calcium, and using the same probe, they found that calcium uptake by mitochondria was reduced. These changes were accompanied by an increase in ER stress protein response genes ATF4, ATF6, and HERP, as well as a general reduction in protein synthesis rate. So overall, they concluded that the mutations in astrocytes in this model of familiar Alzheimer's disease affected 3 things in astrocytes, which is number 1, bioenergetics, number 2, calcium homeostasis, and number 3, proteostasis, which likely overall promotes homeostatic dysfunction in astrocytes and the neurons that these astrocytes are communicating with. Okay, our next paper is Serum Amyloid A inhibits astrocyte migration via activating P38 MAPK. First author Lin, last author Yu, and this was published in the Journal of Neuroinflammation. So this study explored the mechanisms of one of the hallmark characteristics of Alzheimer's, which is the aggregation of astrocytes around plaques. This study looked specifically at a protein called serum amyloid A, or SAA, which is known to co-localize with senile plaques, and they explored its role in astrocyte migration. The authors crossed a transgenic mouse line called APP-PS1 with SAA3 knockout mice, and then used Western blots and immunostaining to look at SAA expression and astrocyte activation around plaques. For the effects on astrocyte migration, they used a scratch assay and immunostaining. So these authors found that SAA expression was higher in the aged APP-PS1 transgenic mouse line, and that deficiency in SAA resulted in increased astrocyte activation around plaques. Astrocyte migration was inhibited by SAA in culture, which was mechanistically shown to be a result of disruptions in various functions like actin and microtubule reorganization. They also reported that the p38 MAPK pathway, when inhibited, resulted in suppression by SAA on astrocyte migration. So overall, the take-home message here is that an increase in SAA in the transgenic model of Alzheimer's inhibited astrocyte migration to plaques via the p38 MAPK pathway. Okay, so our next astrocyte paper, and actually the last astrocyte paper, is titled Amyloid Beta Regulates Gap Junction Protein Connexin 43 Trafficking in Cultured Primary Astrocytes. The first author is Malik, and the last author is Sarma, and this was published in the Journal of Translational Medicine. So connexin proteins are gap junction proteins, which are in part expressed in astrocytes. Connexin 43 is particularly associated with neurotoxicity in Alzheimer's disease. So this has already been well established that amyloid beta plaques will induce an increase in connexin 43, also known as CX43, in astrocytes, though the mechanisms of how this works are unexplored. So this paper used astrocyte cultures to further figure out how amyloid beta plaques can change expression levels of CX43. And they found that exposure to amyloid beta plaques impairs gap junction coupling, even though the intracellular pool of CX43 remains high. So this means the dysfunction appears to be at the protein assembly level at the surface of the cell. Intracellular CX3, which is still robustly present, appeared to be retained in the ER-associated cell compartments. And interestingly, the trafficking of newly synthesized CX43 that reaches the Golgi was not affected by amyloid beta plaque exposure in astrocytes. So to support this finding, the authors use a chemical called 4-phenylbutyrate, which is widely used to improve membrane trafficking of proteins. And they found that this treatment restored amyloid beta-induced gap junction coupling between astrocytes. So overall, they concluded that amyloid beta exposure causes astrocytes to rapidly internalize their CX43 gap junctions. So this next paper looks at both astrocytes and microglia in Alzheimer's disease. So it's the perfect segue article as we transition from purely astrocyte-related papers to microglia-related papers. And this is kind of the bridge in between. So the title of this paper is Astrocytic TSPO Upregulation Appears Before Microglial TSPO in Alzheimer's Disease. The first author is Ternier, the last author is Millet, and the journal is the Journal of Alzheimer's Disease. TSPO is a translocator protein, and in vivo PET and SPECT imaging of neuroinflammation actually relies on estimations of TSPO levels. This protein is pretty widely expressed in a variety of cell types, which historically has just made it challenging to kind of parse out the results and interpret them. So the objective of this study was to understand which cells are expressing the changes in TSPO levels in Alzheimer's disease. A couple of radioactive imaging techniques were applied here, including the TSPO radiotransfer and fluorescence-activated cell sorting. And what's cool about this paper is that the authors looked at TSPO levels in both a rat model of Alzheimer's disease, which in this case was the TGF34480 rat line, as well as human Alzheimer's patients. So the authors found that in the hippocampus of the transgenic rats, TSPO expression increases were visible at 12 and 24 months in astrocytes. However, these changes were only visible at 24 months in microglia, so there's some differences there between the astrocytes and microglia expression. While TSPO changes were not exclusive to glial cells in the rat model, in the human patients, TSPO upregulation was found only in glial cells. The authors reported some differences in the mechanisms between the transgenic rats and humans, such as an increase in the number of TSPO binding sites per cell in rats, without any changes in cell proliferation. However, in humans, they found that microglial cell population increased while the TSPO binding site number remained the same.
Hey listeners, I'm here to let you know AMiNDR is recruiting. If you're interested in joining us, shoot us an email at amindrapodcast@gmail.com. You can also find us on Facebook, Twitter, and Instagram. Enjoy the rest of the episode.
Okay, so now we're in the microglia-specific studies, which microglia are my favorite cell type, so we're at the best part of this episode. Our first paper is ubiquitin ligase COP1 suppresses neuroinflammation by degrading CEBP beta in microglia. The first author is Dejoa, the last author is Dixit, and this was published in Cell. Okay, so this study's overarching goal was to further examine the mechanisms that control pathogenic microglia gene expression. CCAAT/CEBP beta, which stands for enhancer binding protein beta, is a transcription factor that is upregulated in Alzheimer's disease and modulates the expression of pro-inflammatory genes in microglia. The study showed that expression of this transcription factor is regulated post-translationally by the ubiquitin ligase COP1. When COP1 is not present, CEBP beta accumulates rapidly and then drives pro-inflammatory gene expression in microglia along with neurodegenerative-related genes. This is partially based on microglia neuronal co-cultures, where absence of COP1 increased neurotoxicity. The study also used antibody-blocking assays to demonstrate that the neurotoxicity reported was basically exclusively attributable to complement. Interestingly, removal of a single CEBP beta allele prevented the pro-inflammatory response from microglia, so COP1 deficient microglia also showed a rapid increase in tau-mediated neurodegeneration. And this altogether suggests that COP1 is critical in microglia for suppressing the pathogenic CEBP beta gene expression. Our next microglia-related paper is titled Penicillin 1 Phosphorylation Regulates Amyloid Beta Degradation by Microglia. The first author is Lido, the last author is Greengard, and the journal is Molecular Psychiatry. So this paper studied penicillin 1, or PS1, which is a subunit of the enzyme responsible for cleaving the amyloid beta peptide. PS1 has been primarily studied in neurons so far, but this study looked at PS1 in microglia and found that microglia which were deficient in PS1 had a slower response time to injury in the brain. This was done in vivo, and they were also unable to degrade amyloid beta oligomers because of impairment in phagocytosis. So using a mouse model of Alzheimer's disease, this paper also showed that is when PS1 is mutated, microglia contain severe amyloid beta aggregation as well as accumulation of PSD95, which is the postsynaptic density protein. Overall, this paper showed a new mechanism by which PS1 can alter microglia health and contribute to the pathogenesis of Alzheimer's. Next up, we have a paper titled Microglia Depletion Diminishes Key Elements of the Leukotriene Pathway in the Brain of Alzheimer's Disease Mice. The first author is Michael, last author Agner, and the journal it was published in is Acta Neuropathologica Communications. So leukotrienes are a family of inflammatory mediators and are known to contribute to neurodegenerative disorders generally, not just Alzheimer's specifically. Because of their role in inflammation and neuronal cell death, when blocking leukotrienes, it reduces pathology and improves cognitive function in animal models of neurodegeneration. This paper aimed to characterize the source of leukotrienes in the brain, which is previously unknown. And the authors identified an enzyme called 5-LOX, which is found in neurons and also in a subpopulation of microglia. To do this, they used both brain sections of Alzheimer's disease patients and tissue from a mouse model of Alzheimer's disease. Their conclusions were that the 5-LOX activating protein FLAP, which functions to anchor 5-LOX to the membrane, is exclusive to microglia. The entire microglia population expressed FLAP, and when microglia were diminished in the brain of the transgenic Alzheimer's mice, the result was a drastic reduction in 5-LOX mRNA expression in the brain and its protein expression in neurons. So overall, this study concluded that microglia are responsible for A, leukotriene biosynthesis, and B, microglia also regulate 5-LOX in neurons. Our next paper is titled Microglia is associated with p-tau aggregates in the olfactory bulb of patients with neurodegenerative diseases. First author Carmona Abelin, last author leukotriene, and this was published in the Journal of Neurological Sciences. This paper investigated the olfactory bulb, which is the structure in the brain that is first affected in virtually every neurodegenerative disease, Alzheimer's disease, but also Parkinson's and dementia. This study aimed to investigate how activated microglial cells contribute to the increased amount of protein aggregates, which appear in the olfactory bulb over the course of all of these neurodegenerative diseases. They quantified the number of activated microglia and also astrocytes in histological brain sections from patients with diagnoses of Alzheimer's and these other neurodegenerative diseases. Olfactory sections from the subjects were immunostained, and the number of activated microglial cells and astrocytes were found to be increased in patients with neurodegenerative diseases. However, these activated microglia did not correlate to the number of beta amyloid plaques or other protein deposits, though they did correspond with phosphorylated tau aggregates. Okay, moving on. Our next paper is titled Attenuation of the Extracellular Matrix Restores Microglial Activity During the Early Stage of Amyloidosis. First author is Stoyanov, the last author is Dityatyev, and the journal this was published in is Glial. This paper aimed to understand if microglia function is altered in the early stages of Alzheimer's disease, meaning so before plaques appear and microglia show their activated phenotype. Here, the authors study the retrosplenal cortex, or the RSC, in young, so meaning 3 to 5 month old, 5 times FAD mice. This time point is early enough in the disease progression that gene expression of genes encoding for the major extracellular matrix proteins in the RSC are not dysregulated yet. The authors of this study found that even in this early time frame of the disease, histological analysis of the fine structure of the extracellular matrix showed an increase in levels of Wisteria floribunda agglutinin, labeling of holes in perineuronal nets, and changes to the parameter of the extracellular matrix barriers. These authors used 2-photon vital microscopy to examine microglia in the 5 times FAD mice, and found that while microglia showed a normal resting morphology, there was a significant reduction in the number of microglia that migrated to a site of damage from photolesion. These effects were alleviated by enzymatic digestion of the ECM by chondroitinase ABC, which is known as ChABC, and ChABC treatment also reduced the invasion of myeloid-derived cells into the RSC of 5 times FAD mice. The authors concluded that microglial and myeloid migration was altered in the early stages of the disease, and that both of these effects can be diminished by reducing the ECM. Okay, another microglia paper we have today is titled Brain Tocopherol Levels Are Associated with Lower Activated Microglia Density in Elderly Human Cortex. First author DeLu, last author Morris, and the journal is Alzheimer's and Dementia Journal. This paper investigated tocopherol levels in the brains of 113 deceased participants. It is already known that higher levels of tocopherol in the brain are associated with lower levels of Alzheimer's dementia, but the mechanisms of this are unknown. These authors use linear regression analysis to explore the relationship between tocopherol levels and microglia density, and they also adjusted for age, sex, education, apoprotein E genotype. And then in a second model, they additionally adjusted for total amyloid load and neurofibrillary tangle severity. What they found was that increased levels of alpha and gamma tocopherol levels were associated with lower levels of activated microglia and lower total number of microglia. This effect remained after adjusting for the elements of neuropathology, and the authors concluded that there is a relationship between microglia activation and tocopherol levels. Next up, we have microglia implicated in tau pathology of the striatum of neurodegenerative disease patients from genotype to phenotype. First author Li, last author Zhu, and the journal this was published in was the International Journal of Molecular Sciences. This paper looked at the interactions between dopamine and oxidative damage in the striatum, which is a phenomenon seen in basically all types of neurodegeneration. Specifically, these authors were interested in how this impacts microglial phenotype. So the authors chose to look at and then quantify levels of 3 different factors, which are MPO, or myeloperoxidase, ADP ribose, and triggering receptors expressed on myeloid cell 2, which is TREM2, using ELISA assays. These authors also looked at the relationship between microglia dystrophy and tau pathology, and they did this by using a translocator protein, so TSPO, and tau fibrils using autoradiography. They did a remarkable job of looking at brains, specifically the caudate and putamen of patients that had Lewy body diseases, which is Parkinson's disease dementia, and dementia with Lewy body, as well as Alzheimer's disease. They genotyped both disease and control brains for the factors I mentioned earlier, so TSPO, TREM2, and then also bridging integrator, or BIN1, genase using single nucleotide polymorphism assays, or SNP assays. The results of this study were that TREM2 gene variants were absent across all samples, but they did find associations between TSPO and BIN1 gene polymorphisms and TSPO. MPO, TREM2, and PAR variants were present. So just to quickly summarize those variations, PAR levels were lower in the caudate of Lewy body diseases. TSPO density and tau fibrils were lowered significantly in the striatum of Lewy body diseases, but were increased in Alzheimer's disease. So overall, the main take-home message of this paper was that microglia dysfunction conversely reduces tau propagation. Okay, so we've arrived at our final microglia-specific paper, which is titled Gamma Secretase Cleavage of the Alzheimer Risk Factor TREM2 is Determined by its Intrinsic Structural Dynamics. First author of this paper is Steiner, the last author is Hagen, and the journal was the EMBO journal, so Excellence in the Life Sciences journal. Like the last paper, this paper also looked at microglial expression of TREM2, which, just to remind you, stands for triggering receptor expressed on myeloid cells 2. Sequence variants of TREM2 are a risk factor for late-onset Alzheimer's disease. In order to function properly, TREM2 needs to have a stable interaction with DAP12 in the membrane for signaling processes. This interaction is terminated by TREM2 ectodomain shedding and then membrane cleavage by gamma secretase. This study aimed to understand the mechanisms and specificity of this intramembrane cleavage event, and approach this question by determining the structure of the TREM2 transmembrane helix, or the TREM2 TMH. What they found is that the TREM2 TMH adopts a kinked structure with increased flexibility due to a charged amino acid in the membrane region, and then removal of this charge leads to TMH stabilization and reduced dynamics, which is similar to the stable complex formed when TREM2 interacts with DAP12. The authors found that this dynamical site matches with the site of the initial gamma secretase cleavage event, suggesting that a yet-to-be-characterized cleavage mechanism involving gamma secretase relies on the flexibility of this TMH region for cleavage specificity. Okay, so that wraps up the portion of this episode on microglia-specific papers, and now we're moving into the final section or category of today's episode, which is general neuroinflammation and immune function in the progression of Alzheimer's disease. This next paper is actually probably my favorite paper of this whole episode, and it's more of a methods paper, but it characterizes a really cool technique that I think will have a lot of application in the future. So the title of this paper is Acoustofluidic Assembly of 3D Neurospheroids to Model Alzheimer's Disease. The first author is Kai, the last author is Gao, and the journal it was published in was Analyst. Okay, so it's really well known at this point that neuroinflammation is involved in the progression of virtually every neurodegenerative disease, which includes Alzheimer's, but it's challenging to fully model the role of inflammation in these diseases. This paper took an interesting and unique approach for disease modeling by using an acoustofluid method that constructs 3D neurospheroids and inflammatory microenvironments in a petri dish. Specifically, this paper was interested in looking at the model of microglia-mediated inflammation and microglial migration. This acoustic assembly is contactless and label-free and is used in cell culture. This method assembles dissociated embryonic mouse brain cells into hundreds of 3D neurospheroids in a hydrogel within minutes. And what's really fascinating here is that these neurospheroids are uniform and they contain controlled cell numbers and composition. And by composition, that means the ratio of neurons to astrocytes to microglia. And they can also control environmental components. So in this case, they added in amyloid beta aggregates. And using this approach, these authors were able to maintain and monitor the interactions between cells and plaques in real time for a time span of days to weeks, using high-throughput time-lapse cell imaging. And what's really helpful and accessible about this technique is that it works with a simple setup. So the acoustofluidic technology works with conventional petri dishes and well plates. Okay, so our final paper in today's episode is about the glymphatic system and its role in clearance of proteins such as amyloid beta plaques and tau, which are of course really relevant in the disease progression of Alzheimer's. So the title of this paper is Impaired Glymphatic Function and Clearance of Tau in an Alzheimer's Disease Model. The first author is Harrison, the last author is Lythgoe, and this was published in the journal Brain. The specific goal of this study was to look at tau protein transference via the exchange of cerebrospinal fluid with interstitial fluid, which is the clearance system for tau that appears to be dysregulated in Alzheimer's disease. The authors found that there is a dysfunction in the CSF-ISF exchange and that aquaporin 4, which is a channel involved in the CSF-ISF exchange, appears to be impaired in a mouse model of tau pathology. They concluded that aquaporin 4 is central for the process of glymphatic clearance of tau accumulation and that the CSF-ISF system represents a novel drug target for the treatment of Alzheimer's disease. Okay, and that brings us to the end of today's episode. Thanks so much for tuning in to this episode of AMiNDR on the role of glia, the immune system, and inflammation in Alzheimer's disease.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, yeah, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.